What is transcription? So if you think about like the act of transcribing, right? So if your job was to be a transcriber, then you would listen to me talk and write it down. Transcription is the same, it's a similar thing. It's reading the code in DNA and putting it in an RNA language, okay? So you're transcribing the DNA into the RNA language so that it can leave the nucleus. So under normal circumstances, DNA is confined to the nuclear area in a eukaryotic cell or in a nucleoid region in a prokaryotic cell, and the DNA resides there always, okay? But if you need to make a gene, so if you needed to transcribe a gene, if you needed to express it, if you needed to make those proteins, then you had to go in and read the DNA and then carry the message outside, okay? So RNA is allowed to leave the nucleus, DNA is not. So we have to transcribe so that we can get DNA's message out into the cellular world. So how do we do it? Probably a little less complicated than replication because it's not as many players. So here in blue, you see your DNA molecule. And here in red, you have your mRNA molecule that's being newly transcribed. Now, you don't have to worry about all the jobs of replication. Here, the polymerase does everything, okay? So it causes the DNA to unwind, it reads the code to add those new nucleotides, and then it allows the DNA to rewind behind it, okay? So the DNA is only open for a short while so that message can come out, and then it closes right back up again. So in transcription, you have this RNA polymerase now that comes and attaches to the DNA molecule, opens up that DNA molecule, reads it, adds those nucleotides to form the RNA, and then lets that DNA close back again, okay? Similar things as replication, you have your orientation of your 3 to 5. Your RNA also has an orientation. The RNA molecule that you're made is being read from a template strand, okay? That's the strand of DNA being used for transcription. The other strand, the non-template strand, is going to be the same as that mRNA molecule because we remember about the complementation. So if this was an A and this was a T, when we read this, we're going to put an A on that RNA molecule. Okay, so you will see an A and an A. The only difference would be that instead of having thymine in this red molecule, you're going to have uracil because that's what goes in RNA, right? We remember that. And so then another thing will be what happens after you make that RNA transcript. Now that transcript can then leave the nucleus, and it could be made into a protein or it could do other things. So you remember one of the first pictures we looked at was a ribosome and a tRNA molecule. Those are made the same way. This here is an mRNA molecule. It's going to leave the nucleus and be made into a protein. So in order to do that, we have to translate that message, okay? So first we transcribe from DNA to mRNA, and now we're going to translate from an mRNA to a protein. So translation is the second half of that, and it's taking that mRNA molecule and making that protein. It's necessary because this is how we make our proteins. And we all know where we make our proteins, right? So where does translation occur? Correct, at the ribosome. So here's our beautiful ribosome, whose job it is to make proteins. This ribosome can be found in that rough ER or in the cytoplasm hanging free. So this messenger RNA, this mRNA molecule, finds this ribosome. You have these tRNA molecules that have an amino acid at one end, and we know that amino acids are the building blocks of our proteins, okay? And then this ribosome is our RNA. So all of these molecules will be transcribed, but only this mRNA is translated. So there's 3 positions on a ribosome. So an E, P, and A site. The A site accepts those incoming tRNA molecules. The P site is where the peptide bond is formed, and then the E site is where it leaves. So some ribosome pictures show 3, some show 2 sites because there's still some discrepancy over whether the E site is really a site or not. Okay, so it comes into the complementary. So everything that we do with DNA and RNA is based on complementation. So this is UGG. The complementary pairs will be an ACC. So you see the ACC. So when it comes in, it has to bind to its complementary pairs. It binds to those pairs, it shifts, the peptide bond forms between the two amino acids, and it's going to shift again, kick one out, and it has an open space to bring one in. So you have an open space. The tRNA molecule that's complementary to this is an anticodon. It's a codon on the mRNA. That's a three nucleotides. So the tRNA that's complementary to the mRNA codon comes in and binds. The RNA is going to shift, kick out one of the tRNAs, leave space for another one, and form a peptide bond between the two that remains there. Okay, so think of this like an assembly line. When it's your turn, you come in carrying your amino acid. Once the space opens up in front of you, you'll move ahead. You'll form a peptide bond with the amino acid at your top next to your neighbors, and then the space opens for you to leave, and you can go and get another one. So imagine walking into a room carrying a ball. The ball is going to represent the amino acid. Every time there's a shift change, you can move up one step. When you're in that P site, you lose your ball because the other guy is going to collect it and form that peptide bond. You're going to go out of the room and try to find another ball so that you can come in when it's your turn to be complementary again. And this goes on and on until we reach an exit. And then everything falls apart so it can be recycled again so that this process can continue to make all the proteins that you need in the body. So now, if I give you a DNA strand, you know this is DNA because you see T's in it, okay? If this was an RNA strand, you would see U's. So if I give you this DNA strand, you would give me the complementary mRNA strand, okay? So now, where you see a T, you're going to put a U, right? Where you see an A, back to those T's, your C's go with G's, and then you would continue this process. Now, after you get the entire strand, the next thing I would tell you to do is mark off, this is an A. Mark off every 3 because that's your codons. Remember we talked about codons? And then you would do it up here as well, okay? And then you have to figure out what is that amino acid sequence. Well, in order to do that, you need this codon table. So we said that this was an ATG. And so you will come here to the table. So first letter here is an A. Second letter, is a U. And third letter is a G. So you're looking for AUG. So here's AUG. It's MET. So if you go back then to your sheet, you're going to write MET because that's the next one. Now you see CCC, which is complementary to GGG. Go back to your table, find GGG. G, first letter, G, second letter, G, third. So GGG is glycine. Go back to your sheet and you'll write down glycine. And you'll continue that for the rest of your DNA until you get your complete protein. So once you get that amino acid sequence, that's going to be the protein sequence that you're going to create, okay? Now, so when you're doing this, I want you to think about this as a message. So when you go to the library, there's certain books that you can't check out. These are called your reference books, right? They have to stay in the library. Well, how can you get that message if you really need that book for class? So you have to go to the library, which is going to be analogous to our nucleus. And that book that you need is going to be similar to our DNA. Now, you can't take it out, but you need the message. So you're going to photocopy it because those photocopies that have the similar message, it's identical in this case, but it's still a photocopy. It's not the original, can leave the library, okay? So you go in, you make your photocopy, and then you carry that photocopy out with you. That photocopy is going to be your protein. And so that's what I want you to think about in this case of how you make or how you utilize that DNA to make that protein. So your nucleus is represented by your library. There's a reference book that you can't take out of the library, which is representative of our DNA. So you make a photocopy, which is going to be similar to what RNA does, and then you can carry it out of the library, take it home, and use that information because it's your protein. Okay, so hopefully that helps here. And then depending on what it is, in this case, it's you're going to translate it into some product, and this product will be the recipe if you made the product.